Okay, so we already extracted the header information from one of the other files. So can we do it again for this file and display it? Yeah, it's kind of similar. Number of channels, 64 is 160 hertz sampling. Subject info, 64 channels. Low pass, high pass, that's fine. 80 hertz should be sufficient enough. Anything below 80 hertz should be good enough, but it doesn't tell us what the file actually is. So we can view it and stuff, but yeah, let's listen to that 8 minutes and then keep going.
We're diving into the real scientific data, specifically EEG data, that's electroencephalography, from the PhysioNet database. And, it's never just about downloading files, is it? It's more like peeling back layers, understanding the quirks, the details, and sometimes hitting unexpected roadblocks with real-world data. It's about finding the story in those signals.
Absolutely, and that's what we want to illuminate for you today. We'll walk through the kind of practical steps and, the common hurdles you hit when you acquire and first analyze this type of data. Think of it as maybe a shortcut to getting a handle on biomedical data. You'll see how to approach it, what questions pop up, and why tiny details like, say, file formats really matter a lot.
Okay, so where did we actually start? The beginning of the trail. It kicked off, like a lot of data projects, with downloading. Pulling a specific EEG dataset from PhysioNet, that public repository, and we used regget for that, the command line tool.
Yeah, regget. Simple enough to get the files downloaded. But then the really critical first step, once you have the files, is inspection. We used the mine library in Python. It's really the standard for neurodata. We used it to load the data and, crucially, just look at the header information first.
Okay, the header. What were the first key takeaways from that? What did it immediately tell us?
Right away, several things jumped out. It showed 64 channels.
64, okay.
Sampled at 160 hertz. So 160 data points per second per channel.
Which is decent for EEG, right?
Oh, yeah. 160 hertz is pretty solid. Captures brain activity well without being, excessively huge data-wise. The header also confirmed the channel types were EEG, as expected. But interestingly, it noted that head sensor digitization wasn't available.
And what does that mean? Practically no digitization.
It means we don't have the precise 3D coordinates for where each electrode was placed on the head,
right?
Which, it limits certain types of analysis later on, like detailed source localization. It's a common thing with real-world data, actually.
So right from the start, you get a sense of the data's shape and maybe its limitations.
Exactly. That initial header info is foundational. It sets the stage for everything else.
Got it. Now, you mentioned getting the files.
Yeah.
But it wasn't just one single EEG file, was it? Usually isn't.
No, definitely not. When you download these datasets, you often get a whole collection of things, supporting files.
Like what else came along with the EEG data?
Well, there were a few interesting ones. There was a 64-channel Charbot.png.
Okay, .png, so an image file, probably showing the electrode layout.
Almost certainly. A visual map of those 64 channels. And there was a PDF version too, 64-channel Charbot.pdf. That could be a more detailed document, maybe specs, maybe notes on the setup.
Makes sense. What else?
Then the really important bit, a directory named records.
Records. That sounds like where the actual data lives.
Precisely. That's where we found the core EEG data files in EDF format, European data format.
EDF, right? Common for clinical and research EEG.
Very common. We also saw a file called SHHA256 semss.txt.
Checksums. For verifying the download wasn't corrupted.
Exactly that. Super important for data integrity. You run a check, make sure the files you have are identical to the ones on the server.
Good practice. Anything else catch your eye?
There was one more. WFDB Kale. The name suggests maybe calibration files or perhaps something related to the WFDB toolkit.
WFDB. That's the software library often used with PhysioNet data, isn't it?
Yeah, waveform database. It's a whole suite of tools and formats common on PhysioNet. So WFDB Kale likely fits into that ecosystem somehow.
Okay, so lots of pieces. But for the brainwaves themselves, we needed to look inside that records directory at those EDF files.
Correct. That's the heart of the electrophysiology data.
Right.
But then things get a bit more complex. We found other files alongside the EDFs, specifically event files.
Event files, what are those typically for?
They're critical, actually. They usually contain markers, timestamps, indicating what was happening during the recording, like when did a task start, when did a stimulus appear, when did the subject blink, maybe, things like that.
So they let you sync the EEG waves with actual Events.
Yeah.
Makes sense. Vital for analysis.
Absolutely vital. You need to know what the brain was reacting to.
So did loading these event files go smoothly? I sense a but coming.
Yeah, there's the but. So we took one subject, S014R02, loading their main EDF file with M&E. Worked fine, no issues.
Okay.
And then we tried to read the corresponding .event file for that same recording, and error.
What kind of error?
It was an encoding error. M&E tried reading it as standard UTF-8 text, and it failed.
Not text then.
Well, we tried rereading it specifying a different encoding, Latin 1, which sometimes works for unusual text files. Still no good. Looking closer, the file was full of morabytes, non-printable characters. It wasn't text at all.
Binary data.
Looked like it. A custom binary format, which means you can't just open it like a notebook.
You need to know the specific structure, how the bytes are arranged, what they mean.
Exactly. It requires custom code to parse it. You need the key, the specification for that format.
Okay, so roadblock. How do you tackle something like that? Where do you find the key?
The first port of call, always, is documentation. You hunt around the source. So back to the PhysioNet website for this dataset, the EEG MIDB, looking for any description of the .event file format.
That's the detective work part of data science, isn't it?
It really is. Sometimes the info is right there, sometimes it's buried, or, well, sometimes it's missing.
Did checking other subjects help? Maybe it was just a glitch with that one file.
Good thought. We did try another one. Subject S001, record R01, downloaded those files.
Same issue with the .event file.
Same issue. EDF loaded fine. .event file was unreadable as text, likely binary.
So it confirmed the pattern. These files definitely need special handling.
Right. It wasn't an isolated problem. We knew we'd need to figure out this custom format or find a tool that already knew how.
And this is where maybe that other library comes in, the WFDB one you mentioned. Could that help?
Potentially, yes. The WFDB Python library is specifically designed for PhysioNet data. While it's often used for reading signal data or standard annotations, sometimes header information read via WFDB.R header might contain clues. Or maybe the library has functions specifically for related annotation or event formats used on PhysioNet. It's another avenue to explore.
It really shows how iterative this process is. Download, try to read, hit an error, search for docs, try a different tool.
That's exactly it. It's rarely a straight line from download to analysis, especially with complex real-world scientific data. You poke, prod, troubleshoot.
And just to circle back for clarity, that SHA-256SUMS.txt file, the checksum one, that doesn't help with understanding the event file content, right? It's purely for checking if the file downloaded correctly.
Precisely. It verifies integrity, that the file hasn't been corrupted. It tells you if the file's okay, but absolutely nothing about what's inside it or how to read its structure.
Got it. So two different kinds of problems. File corruption versus unknown file format. And the checksum only helps with the first one.
Correct. Parsing the .event file requires understanding its internal structure, its specific language.
So what started as getting EEG data led us through this landscape of company files, images, PDFs, checksums, and right into this critical challenge with the .event files.
Yeah, and it's such a great illustration of working with scientific data. It's not just about the big numbers or, the cool brain plots. So much of it is this meticulous groundwork, understanding the file structures, paying attention to detail, and doing that detective work when things inevitably don't just work out of the box.
And that aha moment, realizing those tricky .event files are the absolute key. Without them, the EEG is just signals in a void. They connect the brain activity to the real world, to what was actually happening. That's where the meaning comes from.
Exactly. The raw signal tells you that the brain did something. The event file tells you why, or at least when in relation to external occurrences. This whole process, trying to crack open these data formats, it really feels like a small version of scientific discovery itself. It makes you pause and think, doesn't it? How much incredibly valuable knowledge about our brains, our bodies, the universe might still be locked away, hidden inside complex data sets with formats that are obscure or undocumented, just waiting for someone to figure out how to read their hidden language.
Okay. There's a human in the loop. Seem to be struggling. Might need a prompt engineer again. Where's that prompt engineer? I might actually look for other datasets. Okay, what's happening on the screen at the moment?
The screen is showing a Python coding environment, likely a Jupyter Notebook or Google Colab. There appears to be code related to processing EEG data, possibly involving loading channel montages and calculating frequency bands.
Again, what's the problem? what's the current problem is? Can you explain in detail?
The main issue is that the channel names in the dataset don't perfectly match the standard montage being applied, causing errors. Also, there was an error in calculating the frequency indices, which needs to be corrected.
Okay, so can you act as a prompt engineer and instruct this developer as to what to do? But keep in mind, this is meant to be a highly experienced developer, so just perhaps ask them some detailed questions.
To ensure successful processing, let's focus on a few key areas. Could you verify the exact channel names expected by the standard montage and ensure the renaming step aligns them correctly? Also, please double-check the logic for calculating the frequency indices to ensure they accurately capture the desired band. Finally, let's confirm that the Topomap plotting function is receiving the correct channel location information. Can you provide more details on how these steps are currently implemented?
Yes, so can you overview the response from the developer? There seem to be a lot of issues. Can you respond to them directly? So review the response and respond to the developer.
Certainly. Regarding the channel renaming, it's crucial to ensure the exact match with the standard montage. Double-checking the current names against the expected format will help resolve the discrepancy. For the frequency index calculation, ensure that the band limits are correctly identified and that the indexing method correctly extracts the relevant frequencies. Lastly, verify that the Topomap function is receiving accurate channel location data from the corrected channel montage information. Addressing these points should help resolve the plotting and indexing errors.
Okay, and can you review and suggest, can you give an overview of what we are doing at the moment?
Currently, you're working on processing EEG data, specifically trying to apply a standard montage and plot a topographical map. The main challenges involve resolving channel name mismatches and ensuring accurate frequency index calculations to correctly plot the topomap.
So whatever we do is eventually available on this website. Can you give a quick overview of what the website is about?
The website appears to be focused on biomedical technology and education, offering tools and interactive resources for researchers, educators, and students in fields like EEG data processing. It seems to provide modules for simulating data, exploring medical data landscapes, and guiding interactive EEG technologies.
Okay, what's currently happening development-wise?
The developer is currently refining the code to address the channel name mismatches and frequency index errors. They are also modifying the code to ensure the Topomap plotting function works correctly with the updated data. The focus is on accurately mapping the channels and providing the correct location information for the plot.
Okay, can you ask the developer a question?
Could you provide more details on the specific channel names that are causing mismatches and how you are currently renaming them? Understanding the naming convention differences will help in pinpointing the exact issue.
Okay, so we have these names, we have this montage. Can you explain it briefly?
The montage is a specific layout of EEG electrodes on the scalp, defining their positions according to a standard system. It's crucial for correctly visualizing the electrical activity of the brain and ensuring that the channel data aligns accurately with their physical locations on the scalp, in plots like the Topomap.
Okay, and we seem to be having all the names in this specific file. So can you ask the developer?
Can you confirm whether the channel names listed in the file records.txt correspond exactly to the channel names used in the standard montage? Also, are there any specific channels that are consistently causing mismatches when applying the montage?
Okay, what's happening? What's happening? Oh, okay, we get a montage. So that's good progress.
Yes, it looks like the Topomap plot is now displaying correctly, which is a significant step forward. The key was ensuring the channel names match the montage and handling the color bar appropriately. This indicates that the plotting functionality is working as expected. It seems there's an issue with accessing the kind attribute of the montage object. This could be due to changes in the library version or an incompatibility with the specific montage being used. You might need to adjust the code to access the necessary information differently. Based on the error and the displayed file names, it seems the code is trying to access a file named WFDB cal.txt, which doesn't exist. You should instruct the developer to ensure the file path is correct and that the file records.txt is being referenced properly for the channel names. The current issue is that the code is looking for a file named WFDB which it cannot find, causing a file not found error. Additionally, there's an attribute error indicating that the montage object does not have the attribute kind, which suggests an issue with accessing the properties of the montage in the current library version. Now that the Topomap plot is working, the next steps could include event extraction and epoching the data based on experimental tasks. You could also consider artifact detection and removal to improve data quality, and then proceed to time frequency analysis or evoked potential analysis to study brain activity related to the experimental stimuli. Based on the information provided, the dataset mentions motor imagery tasks. However, specific details about the exact stimuli or event timings for evoked potential analysis would typically be found in the dataset documentation or the accompanying event files. You'll need to refer to those sources to get the precise information for stimulus-locked analysis. Before abandoning the dataset, it would be worthwhile to thoroughly examine the available event files and documentation for the EEG data. Sometimes event information is stored in different formats or requires specific handling methods. If you can locate and successfully extract the event information, the dataset could still be valuable for further analysis. It appears the code is attempting to read the event file, but it encountered an event with no data, which caused it to be dropped during the epoching process. This could happen if the event marker is placed too close to the beginning or end of the recording, or if the event duration is too short. You might need to inspect the event file more closely to understand the event markers and their timing. The code is now attempting to interpret the binary content of the event file. It's trying to extract event timings and event IDs from the last 8 bytes of the file, assuming a specific data structure. This is being done to understand the format of the event data and how to correctly extract it for further analysis.
Okay, it seems like we need to look to abandon this dataset and look at another one or something. What do you think?
Given the difficulties in correctly interpreting the event data and the potential issues with the file format, it might be more practical to explore alternative datasets that have well-documented and easily accessible event information. This could streamline the analysis process and help you focus on your research questions rather than troubleshooting data format issues.
Just before we move on to a different dataset, this chart here, so this is showing 0 seconds. Have we turned it into an animation where we can scroll or play, the time, the evolution of the recording, the EDF file, brain recording, EEG recording?
Yes, you can create an animation to visualize the temporal evolution of the EEG data. This involves generating a series of Topomap plots over time and then combining them into an animation. This will allow you to serve the changes in brain activity across the scalp throughout the recording or during specific events.
Okay, what's the plan for that?
The plan is to first select the EEG data for the time window you want to visualize. Then you'll generate a series of Topomap plots for small, successive time intervals within that window. Afterward, you can use a library like matplotlib animation or imageio to combine these topomaps into a GIF or video animation. Finally, you can display the animation in the notebook and summarize the process and results.
Okay, what's happening?
The code is now set up to create an animation of the EEG data. It has selected the raw data from the S01R02 file and defined an animation window of the first 5 seconds. The next steps will involve generating Topomap plots for this time window and then combining them into an animation.
Okay, can you explain the code real quick?
The code is generating a series of Topomap plots over time. It first defines the time duration for each plot and then iterates through the specified time window, creating a Topomap for each interval. These plots are stored in a list, and the code ensures that the time window doesn't exceed the total duration of the data. Finally, it prints the number of topomaps generated for the animation.
Okay, what was the error?
The error is a type error, indicating that the topomap figure cannot be unpacked into axes, which suggests there might be an issue with how the figure is being created or how it's being handled in the plotting function. It's likely that the topomap function is returning multiple objects, and the code is expecting only one object, causing the unpacking error.
Okay, and what's happening now? And can you explain the code real quick?
The code is now attempting to generate topomap plots again, but this time it's explicitly setting the scaling factor.
Yeah, keep going.
Oh, it looks like the same type error occurred again. The issue is still related to how the topomap plot is being generated and the way the figure and axes are being handled. The code needs to be adjusted to correctly capture and reuse the output from the topomap function.
Okay, so what are we doing different now?
The code is being adjusted to handle the output from the topomap function differently. It's now removing the axes parameter from the plot topomap call, as the documentation suggests this might resolve the unpacking error when plotting a single plot. The goal is to correctly generate the topomap figures so they can be combined into the animation.
Are we going in the loop?
The code is trying to iterate through the time windows to generate topomap plots, but it encountered a value error. This error indicates that the time range provided for the topomap plot is invalid, specifically that the start and end times must be between 0.1 and 0.1 seconds, which is not possible for a valid time range. It seems there's an issue with the time window calculation within the loop.
Okay, and did we succeed? And what was changed?
Yes, it looks like the animation frames were generated successfully. The issue was resolved by correcting the way the topomap plot was being generated and ensuring that the time window parameters were correctly passed to the function. The code now generates 50 topomap figures for the specified time intervals. The next step will be to combine these figures into an animation.
Okay, that should be easy, shouldn't it?
Yes, combining the plots into an animation should be relatively straightforward using the imageio library. The code should now save each topomap plot as an image file, and then use imageio to combine these images into a GIF animation. It should proceed without any major issues.
So we are live streaming the whole process on YouTube at the moment. Do you think the viewers should like, subscribe, and check the website where we publish everything eventually?
If you're finding this live stream helpful and interesting, please give the video a like, subscribe to the channel for more content like this, and be sure to check out our website for more details and additional resources. Your support really helps us create more valuable content.
Okay, what's happening now? What do we do to rerun this GIF?
The animation has been successfully generated and is now being displayed. If you want to rerun the animation again, you can simply re-execute the code cell that displays the GIF animation. This will refresh the display with the generated animation.
Okay, and can we save the GIF somewhere?
Yes, you can save the generated GIF animation. The code should have already saved it to a file, likely named topomapanimation.gif in the current directory. You can explicitly specify a different location in the saving command.
Okay, we have the file. Can you explain the file real quick? And what did we do, the whole process?
The file is a GIF animation that visualizes the temporal evolution of the EEG data using Topomaps. Each frame of the animation represents a snapshot of the brain activity across the scalp over a specific time window. The entire process involved selecting the EEG data, generating topomap plots for consecutive time intervals, saving these plots as images, and then combining them into a single animated GIF. This allows for a dynamic visualization of the brain activity over time.
And how do we know it's accurate?
The accuracy of the animation depends on several factors, including the quality of the EEG data, the correct application of the topomap plotting function, and the validity of the time window selected. Assuming the code is implemented correctly and the data is representative, the animation should provide an accurate visualization of the EEG activity over time. However, for precise interpretation, it's always important to consider the specific characteristics of the data and the methods used.
And how do we rerun the animation again?
To rerun the animation, you can simply execute the code cell again that contains the display animation command. This will reload the GIF animation and display it again for you.